social time. Hey, Anna.
Hi, Anna.
Hi. What's up? Today I realized this chapter is definitely a nice break from last chapter. I was thinking it was a little bit easier. It is a lot less material and definitely easier on the brain. So we actually don't even have a lot to cover in class today, so the lecture time's not going to be very long. And we're basically going to kind of talk about what we're doing, and then we're going to simulate making a protein. So we're going to simulate doing transcription and translation, which will help those two things kind of come to life. So that's what all those papers are on your table, and so we're going to work in teams. So you're with your team right now. Last time I had everybody do their own, but I think it's just going to be easier to work in teams. And then there's also a lab in here that on page 197 that was the DNA extraction, and I decided it is a mess and kind of a lot of work on me bringing stuff back and forth on that lab. And so I actually decided that we were going to watch it on video. There's a good video of someone doing that lab with strawberries instead of peas. And so I feel like it gives you the gist of it and it kind of simplifies things for us. It takes a little less time. And so we're going to watch the lab. And the nice thing is I'm not going to make you write it up. Yay! I want to be caught up. The activity today, we're not writing up. Okay? Next week, the candy lab, we do write up. So I think there's maybe the candy lab and one more that gets a write up. But I just decided that I would rather have you guys have time to get caught up on your lab notebooks if you're behind, and we're not going to write up today, and we're going to do it online, and that will also help Anna Bentley because she can just watch the video of it and then kind of do the activity on her own. So, did I get all the folders?
Sorry, I'm not watching sexes in
Oh, it doesn't have to be in here.
Oh.
Yeah. Hey guys, the homework or the corrections or anything like that, it doesn't have to be in the envelope. Like a lot of times you might not have those ready to go in the envelope when the envelope needs to get sealed up. So don't ever feel like everything has to be in the envelope. Okay, I only have 4 envelopes. These are the 2 I'm missing. These are 2 I'm missing. Okay. So I really want to emphasize to everybody, I'm proud of Eli because he told me that he's completely caught up for class today. But next week, okay, listen. It's going to be very important next week to be caught up and to have watched the videos because you are going to get so much more out of the candy simulation lab, and it's going to like solidify the information in your head. And this is what we're doing next week. It's not as hard as what we did with the last module, okay? But so I really make sure you watch the videos, make sure you kind of have a general understanding, and then the activity is going to be a lot more enjoyable for everybody. Okay, so this chapter is about DNA. It's kind of like the start of genetics. It's about cell division. It's about making proteins. The next chapter is the one that goes into genetics in full effect. But the chapter kind of starts out discussing factors that determine what we become. Because a lot of people think everything is about genetics, that it's all genetic, that that's what it's all based on. But what other things besides your genes affect what you become?
Your environment, and then also like what you believe in. So like, say you're raised in a Christian home, and Muslim home, or whatever.
Right, so environmental factors and spiritual factors. And, you know, I think when you get so focused on science and genetics, you kind of, people that do that take God out of the equation, and God can do anything and he can change anything. And so we always need to remember that somebody might be born with a genetic condition or a genetic tendency for something, but God can always change that and heal that. So we always want to remember that. But what is a gene? Essie.
The section of DNA that codes for the production of protein or protein, thereby causing a trait.
Right. So our traits, pretty much, there's so many things in our body are controlled by proteins, okay? So the gene, hey, I need it. Girls in the back, I'm gonna have to move off to the front. Okay. So yeah, I want everybody to kind of sit with the people that, I feel like you guys have your people you like to sit with, but if we have to switch it up because we can't focus, then we'll have to switch it up. But there'll be talking time later, so. Okay, so yes, so the gene is the section of DNA that's gonna code for a certain protein that is gonna be responsible for a certain trait. Okay, so how does a gene, that section of DNA, become a protein? Which this is really what today is about. So there's two big processes that have to happen for that gene to become a protein.
Were
Does
they
anybody
in
remember?
synthesis?
Yes, but what has to happen first with the DNA? What would be the first major step? Transcription, and that is, does anybody remember what what happens with transcription? What is getting transcribed? So you're transcribing would be like writing something that's written in one language into another, so.
The information of the DNA, so that's what the RNA does.
Okay, so the DNA is getting transcribed into
The RNA?
the RNA, okay? So RNA is like a kind of like the opposite snapshot of the DNA. And who remembers where the transcription, where does it happen? Where does transcription happen in that cell? Eli?
Is it the nucleus?
Yes. It is the transcription is gonna happen in the nucleus. Okay. RNA. So what does DNA stand for? Anybody remember what DNA stands for?
Deoxyribonucleic acid.
Yes, and okay, and then what does the RNA stand for?
Ribonucleic acid.
Yes. So and tell me some of the first difference, which is noted in the name, is that the sugar in DNA is what? Deoxyribose. And the sugar in RNA is what? Ribose. Okay, so these are some differences. What is one of the other big differences that you would just be able to look if you looked at DNA and RNA? Another one, Ruby.
RNA only has one strand.
So RNA is single-stranded, and what is DNA? Double-stranded. Okay. Okay, so DNA is going to be double. And then, how do, on DNA, just think DNA right now, the bases, okay, which bases, and those bases are connecting in the ladder on the inside, that's what's connecting those two strands together. What bases connect in DNA? And you remember, what are the sayings to remember it?
Trees and apples and cars and garage.
Apples on the tree. Apples on the tree.
I'm saying the same thing.
Cars in the garage. And so, what are the two that connect for DNA? What are the connections, Ruby?
It's thymine and adenine, and then cytosine and guanine.
Okay. So, we know that.
Wait,
I thought that was
isn't it for
DNA.
DNA?
Oh, it is. I was writing it on the wrong
Oh.
side.
I was like, hold on, isn't that yours?
Okay, And so, Essie, what is the difference over here on the RNA side?
Apples under trees, so it has, what's it called? Uracil.
Adenine and uracil pair together, okay? They're gonna stick together, and then,
Car in the garage.
and car in the garage. So
That's
basically,
one.
the T is gonna be changed out for a U, okay? And so I think if that's confusing and you're like, I have no idea what she's talking about, I think when you do the activities today, you'll understand that, a little bit better. So, okay, this is one thing that was mentioned in the transcription video, that was not in the textbook, but it is something that, is important that I expect you to know. I don't think it's in the textbook. I don't remember seeing it in the textbook. But there is an enzyme that gets transcription started. It's not just RNA, DNA is not just going to start transcribing into RNA without this enzyme telling it to get the process started. It was on the video. I don't think it's in the
I
book.
need to start taking notes. 'Cause I watch videos, but then I forget it.
Yes. So this was a, and I didn't know if anybody would know, but do you know Eli?
Is that a nucleotide?
No.
Okay. It was worth a try.
It's not. No.
So this is some kind of enzyme. Okay. It's called, so everybody write this. Do you think you know it, Hannah?
No, I can't find it. I know it, but I can't find
Okay.
it.
Yeah, it is good. So part of the reason I like you to watch the video after the book is because it's like extra reinforcement explaining it, and sometimes they have some extra details that aren't in the book. Now, I don't always expect you to know and understand all the extra details in the video, but this is one that I do want you to know. But the enzyme that's going to get transcription started is called RNA polymerase. Okay? And that is going to get it started. Okay? And so whenever you see this word polymerase, okay? So RNA and DNA are polymers made up of nucleotides. Okay? And so any enzyme that has the name polymerase in it, it's basically an enzyme that gets the process started to make some kind of polymer. So you look at that poly and then erase, it's getting a polymer made. So we're making a strand of RNA that's a copy, kind of a reverse copy, a negative copy of a strand of DNA that's going to code for a certain gene. And then that RNA is going to be used to actually make the protein in the other process that was discussed in this chapter. Does anybody know what that name is?
For
Where
what?
the RNA is going to make the protein. Does anybody know what that would be called?
Messenger RNA?
It is messenger RNA.
Is it tRNA?
There-
It's like
Translation.
Translation. So I'm looking for the big name of the process. So it's going to be called translation. Okay. So when the RNA polymerase goes to that strand of DNA, first of all, that strand of DNA is going to separate. It's almost like those rungs on the ladder of those bases that are attached together. It's going to pull apart, and so you're going to have a section of DNA, two sections that are pulled apart, okay? That RNA polymerase is then going to attach to that section that's pulled apart, and it's going to start the process of transcription, okay? Which is all happening in the nucleus. So, if we have a section of gene that is, erase this. It looks like this. I'm gonna do it this way so you can kind of picture it like a strand. so this is the DNA strand. And the RNA polymerase comes in and gets the process started. And so we're gonna make a negative copy. And what is this strand over here that we're gonna make called? What kind of RNA is it called? Hannah?
tRNA?
No, not tRNA.
Anticodon?
No,
Oh,
Essie?
I was gonna say that.
Not the no. The-
it's the no. Or am I right?
So when we're transcribing DNA, what kind of RNA do we make?
It's in the book.
mRNA.
mRNA.
Oh, that's all
Okay. So we've got the DNA, and this is transcription right here that's going on, okay? And we're making mRNA. Okay, so when we transcribe an A in mRNA, what is it going to become? It's going to become a U, right? Because
Your use takes place of binding.
What about the T?
Okay,
Okay, so it's going to be an A. And what about the G? C. And what about the C? Okay, so this is a very short strand, but now we've got our short strand of mRNA. And where now is that mRNA going to go to make a protein? The ribosome. So it's heading out of the nucleus into the cytoplasm to a ribosome. Okay, and that ribosome could be in two places. Where could that ribosome be? And this is last chapter.
Just in the cytoplasm or on the
Yeah, yes. So it could either be, it could be going to a ribosome on the rough endoplasmic reticulum or in the cytoplasm. Yes. Okay. So, all right, I'm gonna erase. Okay, so now this strand is going to, I'm gonna draw my ribosome. And the ribosome is like a protein workbench. So I'm gonna call it a protein workbench. And it's where proteins are gonna get put together, and the ribosome, the mRNA is going to determine which amino acids get hooked together in which order to make the protein, because everybody remembers what's the monomer of proteins? Amino acids, yes, and there's 20 different amino acids. So the RNA is gonna tell it what order those amino acids are gonna go in. Okay, I'm going to go ahead and put our strand up here, like it's going through. But I'm gonna make it a little longer. Okay, so this is still a very short strand, all right? And so during the process, and this is our mRNA, there's going to be another type of RNA that's kind of floating around out here. Hannah, what kind is that?
tRNA?
tRNA, okay? And tRNA is not going to be a long chain like the mRNA. And the chain of the mRNA is going to be much, much longer. I just did a short one for our purposes. And so each tRNA is going to have what? Each tRNA is going to have 3 bases. And one amino acid. Okay? Alright, hold on one second. we want to do is we want to get a tRNA that's going to, we're gonna look at these in terms of threes, okay? And we're gonna have a tRNA that has 3 base pairs that are going to pair with these 3 base pairs on the mRNA. Okay, so you're kind of looking at it in terms of 3 because each of the tRNA has the 3 opposite that go here. So this tRNA, and there's going to be tons and tons of tRNA, but this tRNA is going to be attracted to this section of the mRNA. Is everybody following me here? Okay, so it's going to turn its little self around, okay, and it is going to then attach, okay, so we've got, what is the U going to be attracted to?
The A. Oh, A.
A, and the A,
To the U.
and the C.
To the G.
Okay, so this is this piece, it's going up here, this is what's going to connect, and this piece, AUG, is going to have a specific amino acid, and we're going to look at a chart to determine which one it is, that it's going to bring up here to the chain, to the workbench.
It starts with an M, doesn't it?
I think
It's a
it's
messenger.
methionine, is it? Yeah,
Oh.
yes. So I think so, hold on, let me see. Yes, okay, so, everybody got a chart, you're gonna keep this, this is the codon chart.
Yeah.
Oh, but it's actually, the codon chart is actually not, because the codon chart is the mRNA. So it would actually be, I didn't do a start codon, so normally you would have that one first, because that would be the start codon, and you'll see that when we do our activity. But if you're going to look up UAC, so you go to UAC, and that one is tyrosine, okay? So that amino acid is going to be tyrosine, okay? But normally, when the protein gets started, the first one is always going to be the mRNA is going to read AUG, which is methionine, which is the start codon. I just didn't do a start codon. Okay, so then we have GAA, and what tRNA is going to connect to it?
Glutamic acid.
Well, hold on one second. So let's make sure, so this tRNA is going to have CUU, okay? But we look up here, GAA, and Essie, what was GAA? Is that what you were looking at?
I said it.
Yes.
Glutamic
Oh,
acid.
glutamic acid, okay? Because this chart, one thing to remember is, and I actually have to remind myself of this every year, this chart gives you the mRNA section, which is called the codon. So that 3-letter mRNA section is called the codon. What is the 3-letter tRNA reverse called?
Anticodon.
The anticodon. So that's the codon. This is the anticodon. But what you have to know for the chart is just the codon. So you're just reading the part of mRNA for the chart. And what did we say this one was again? Glutamic acid, which the abbreviation is GLU. Okay? So we know we're going to have a long chain, a long polypeptide chain to make up this protein, but we are going to have tyrosine and glutamic acid. But this is just this process is going to keep going and going, and then these amino acids are going to bond together with, anybody remember the kind of bond that the amino acids connect with from Module 5?
Was there a peptide?
Peptide bonds.
Yeah.
Peptide bonds. Good. Good. Okay. So yes, there are, if you look at, everybody look at their chart, you will see there is a start codon, which is AUG, and there are 3 different stop codons, okay? And several codons can stand for the same amino acid. So one amino acid might have several codons that it is attached to, more than one. Okay. and there's gonna be 20 different amino acids. Now, sometimes there's a section of the mRNA that maybe is instructions or extra material and it's not needed. And there might be an enzyme that comes along and cuts it out, okay? And does anybody remember what that is called? When there's like a section on the inside of the mRNA that gets cut out during the process of translation?
Why is it not?
And we can
now?
turn, I didn't even have you guys look at the book, but you can see,
Can you say the question again?
Introns.
introns.
Okay.
Introns.
There were two words, and I wasn't sure if it was exons or introns.
Extrons are instructions that might be at the beginning, and those do get cut off too, but the introns are actually parts on the inside, and it just might be extra information that's not needed. It could be some kind of instructions. And we're still really learning about introns and extrons, what they do and why they're getting cut out. So that's still something that we're learning more about. But that is the gist of it. We're gonna watch the video. So we're gonna go ahead and pull our chairs over here and just watch it on my computer, which is gonna show us the DNA extraction from the strawberry. And then we're going to do that today. So,
I'll just
Do we need to
say,
bring our notebooks?
I don't think so. I think you can just kind of chill and just enjoy the video. Yeah.